The microbiome and chorioamnionitis, labor and delivery. Looking at the normal microbiome, the vagina consists of various lactobacillus species to help sustain a healthy environment by competing against unwanted bacteria, producing lactic acid and hydrogen peroxide to lower the pH levels in the vaginal canal. These are also gram-negative rod-shaped species. Here is a picture displaying a healthy vaginal canal and uterus with normal amounts of lactobacillus bacteria present. Microbiome, aka intraamniotic infection, is an infection of the amniotic sac and amniotic fluid that surrounds the fetus in the womb. This condition is usually caused by preterm labor and misplacement of vaginal and intestinal bacteria. During labor, the cervix begins to dilate, thus allowing bacteria to enter the uterus from the vaginal canal, ultimately causing a major infection of the amniotic sac, and can lead to bacteremia in the mother. These are caused by B. streptococcus, Gardnerella vaginalis, Enterococcus, and Escherichia coli. As you can see within this picture, bacteria is entering the uterus from the vaginal canal and is infecting the amniotic fluid surrounding the baby. The mother's blood vessel is shown underneath to display how septic shock can occur within her. Risks of this disease include premature birthing, neonatal sepsis, endometritis, septic shock. Signs and symptoms include local systemic inflammation, maternal and fetal tachycardia, uterine tenderness, maternal leukocytosis, and malodorous vaginal discharge. Intraamniotic infection complicates 40 to 70% of preterm births. Prolonged labor, premature membrane rupture, vaginal examinations, and vaginal infections are the main causes of developing this type of infection. The study was designed to prove that antibiotics improve preterm birth. In this study, women were given different antibiotics that drastically increased the fetus's development in the womb for 11 more days compared to the women that weren't given any treatment. In conclusion, women that received antibiotics for chorioamnionitis were able to stay pregnant longer than the individuals that did not receive any treatment. Antibiotics were able to delay pregnancy by fighting off harmful bacteria, thus allowing more time for premature babies to grow. For future research purposes, a wider range of antibiotics could significantly contribute to a more proactive approach in treating the future cases of this type of infection. Expanding the participant pool and providing detailed information about each participant, such as their DNA, will allow for more accurate and comprehensive data to be pulled for future analysis. During pregnancy, chorioamnionitis is usually caused by preterm labor, which is out of our control. However, regularly taking antibiotics, limiting vaginal examinations after the water breaks, and getting screened for group B streptococcus in the 3rd trimester will significantly help the preventing of the spread of bacteria through the cervix and into the uterus.